Today, I am going to take up the topic properties and structure of DNA molecule. DNA, you know that it is the hereditary material present in all eukaryotes. Then, there are two types of genetic material. One is RNA and the other one is DNA. In all the eukaryotes, without any exception, the genetic material is DNA. In plant viruses and in retroviruses, we come across RNA as the genetic material. Then, what are the properties of a genetic material? It should be able to make its own copies. That is, it must be in the position to self-replicate itself. Then, it should be structurally and chemically stable. That means it must not undergo frequent mutations. It must maintain its stability. Then, capacity to undergo mutation to contribute to evolutionary process. See, without undergoing mutations, there will be no evolution. So DNA also contributes to the evolutionary process by undergoing frequent mutations. Then, it should be able to express itself as seen in the Mendelian inheritance. It must express itself either by making its own copies or by producing mRNA or by controlling our characters, as you have seen in Mendel's laws. For example, the height of a pea plant may be tall or short. That is decided by the genes. Similarly, the shape of a seed, round or wrinkled, like that, the DNA must express itself. So, any molecule to be considered as a genetic material must possess these properties. Otherwise, it cannot be the genetic material. Then, I have told you, in all the eukaryotes, DNA is the only genetic material. Why? Why DNA is so important over RNA? See the differences. DNA means deoxyribonucleic acid. RNA means ribonucleic acid. You know, these are the acids. They turn the litmus paper into red. Now let us see the differences between these two. Coming to DNA, it is less reactive and more stable. RNA is more reactive and less stable. We have seen coronavirus. It is a RNA virus. We have seen how fast it has mutated and produced so many new strains. So RNA contributes for more mutations. So the organisms possessing RNA as the genetic material mutate very fast. Then, why RNA undergoes faster mutations and why RNA is not stable? The reason lies here. See, this is deoxyribose sugar. This is ribose sugar. I will be talking more about them later on. For the time being, remember, in the DNA, deoxyribose pentose sugar is present. Whereas in the case of ribose sugar is present, or ribose pentose sugar is present. The main difference between deoxyribose and ribose sugar is this molecule, this oxygen. I have given numbers here with the red color. If you take a ribose sugar, there are 5 carbon atoms. This is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon. Similarly, in deoxyribose also there are 5 carbon atoms. 1, 2, 3, 4, 5. The main difference between deoxyribose, ribose sugar is, as the name implies, deoxy means one oxygen is less. So when compared to ribose, here one oxygen is less, and that oxygen is present at second carbon position. Here we find OH group, here only H, hydrogen. And this is a highly reactive group. Because of this OH group, at second position, RNA is unstable and undergoes frequent mutations. So this is a very important point. Then in DNA, there is a thymine. So before that, I think I must explain these things first. There are 2 types of nitrogen bases in nucleic acids. They are known as purines and pyrimidines. Purines and pyrimidines. Purines are 2 types again, adenine and guanine. Adenine and guanine. Pyrimidines are thymine and cytosine. This is about DNA. So in DNA, the purines are adenine and guanine. The pyrimidines are thymine and cytosine. Coming to RNA, purines are the same, adenine and guanine. Coming to the pyrimidines, in RNA, cytosine is present, but instead of thymine, uracil is present. So thymine is replaced by uracil in the case of RNA. So because of the presence of thymine, DNA is more stable. And because of uracil, RNA is less stable. This is one of the reasons. Then, DNA is a double-stranded helix. It has 2 strands. See, this is one strand, this is another strand. Whereas RNA is single-stranded. It has only one strand, either this one or this one. When the molecule is a double-stranded one, this double-stranded structure provides more stability, whereas the single-stranded structure has a less stability. That's why RNA undergoes frequent mutations. Then, DNA molecule is much longer, very, very long. It has millions of base pairs. We'll see the base pairs later on. So a DNA molecule is very long, having millions of base pairs. Whereas RNA is very short. It has only thousands of nucleotides, not base pairs, but nucleotides, only thousands. Then, in the case of DNA, the 2 strands are complementary to each other. I will talk about complementary later on. So in the DNA strand, in a DNA molecule, both the strands are complementary to each other. So DNA is more stable. Because of this complementary nature, DNA is not easily destroyed by heating. We have seen the Griffith's experiment, transformation experiments. He subjected to S-type to heat, but the DNA was not destroyed, and it was picked up by R-type, and it became lethal. Whereas RNA is easily destroyed by heating. So heat denatures RNA, but DNA is not denatured by heat. Then, because of the complementary nature of DNA, the mutation rate is very low. But in RNA, because it is single-stranded, and because of this OH group, RNA is unstable and undergoes frequent mutations. That means the rate of mutation is very high. Then DNA cannot transcribe itself. It cannot participate in protein synthesis on its own. It requires RNA of different types to participate in protein synthesis. But there is no such problem for RNA. RNA undergoes simultaneous transcription and also translation. It itself is an RNA. It requires no further RNAs to participate in protein synthesis. So these are some of the important differences between RNA and DNA, and it also shows why DNA is the preferred genetic material over RNA. For this reason, in all the eukaryotes, only DNA is the genetic material. Then, now let us take up the structure of DNA. The structure of DNA was given by two scientists, James Watson and Francis Crick, in the year 1953. Then, by the year 1953, all the evidence was available to draw a structure of DNA molecule. Among these proofs and evidences, there is one important rule known as the Chargaff's rule. In late 1940s, Edwin Chargaff made one very important rule. Chargaff's rule tells that in a DNA molecule, the amount of purines is always equal to the amount of pyrimidines. Purines are always equal to the pyrimidines. Then, adenine is always equals to thymine. Guanine is equal to cytosine. Then, adenine and guanine together are equal to the amount of thymine and cytosine together. This is called the Chargaff's rule. This is very, very important. So, based on the Chargaff's rule and other evidences, Watson and Crick in 1953 proposed a DNA structure which has revolutionized the molecular biology, and a number of new branches came into existence. Now we are hearing frequently about biotechnology, genetic engineering, DNA fingerprinting, etc. All these have roots in the structure of DNA. Without knowing the structure of DNA, these sciences would not have evolved. Now we have reached a level of creating a new life by using the structure of the DNA and its properties. Now, what is the structure of DNA proposed by Watson and Crick? According to them, the DNA molecule is a double helical structure. This one. Actually, a double helical structure means it is a helix like this. Double helix structure. This is one strand. This is another strand. They have coiled around each other. So this is the double helical nature of a DNA strand. Then these two strands, they run anti-parallel. That means if this strand runs like this strand runs like this. I will explain that later on much more clearly. So the two strands run anti-parallel to each other. But there is also a direction. They run in 3,5 direction. They run in 3,5 direction. I will show you later on more clearly. Then, what are the components of DNA? A DNA has three main components. They are a nitrogen base. So these are nitrogen bases. Any one of the four will be present. Then deoxyribose sugar, this one, and the phosphate group, that means PO4. Phosphate group means PO4. So these are the three main components of a DNA molecule. What are they? A nitrogen base, deoxyribose sugar, and phosphate molecule. Then, how these three structures are arranged? In this figure, I have shown the arrangement of all these three substances in the DNA molecule. These are nitrogen bases. This is one strand up to here. This is another strand. Now let us take up one strand first. Let us take up this strand. These are the nitrogen bases. T means thymine, A means adenine, C means cytosine, G means guanine. I have drawn all the four nitrogen bases. These nitrogen bases are joined to the pentose sugar, deoxyribose sugar. This one. This is the deoxyribose sugar. This is nitrogen base. This is sugar, nitrogen base. Sugar, nitrogen base. Sugar, nitrogen base. By a bond known as glycosidic bond. So, the nitrogen bases are attached to the deoxyribose sugar by a bond known as glycosidic bond. These are glycosidic bonds. Now, we must study the structure of deoxyribose sugar. I have told you, deoxyribose sugar is a pentose sugar. It has five carbons. I have indicated the presence of the carbons with the numbers. This is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon. Then, this numbering is very important. Without knowing this numbering, you can't understand the structure of a DNA molecule. Here I have identified the same. 1, 2, 3, 4, 5. At the third place, and one more point I have to explain here, the pentose sugar, deoxyribose sugar, is a ring compound. This ring compound is heterocyclic in nature. Ring compounds are two types, homocyclic and heterocyclic. If the entire ring is made of a single element, it is known as homocyclic. For example, take benzene. In the benzene, 6 carbons are present in the ring. Only one type, only carbon. But here, you find oxygen also, like this. In all the molecules, oxygen will be present. That means, in the ring, there are two types of elements, carbon and oxygen. Because the ring structure has 2 or more, here only 2 elements, it is known as heterocyclic ring. So, deoxyribose sugar is a heterocyclic ring or compound. So this is heterocyclic. At third place there is OH group. Now, the nitrogen base and deoxyribose sugar and this is the phosphate group. The pentose sugar is attached to the phosphate group on the other side. So on this side it is attached to the nitrogen base, and on the other side it is attached to the phosphate group. We will take up this one. This is the phosphate group, this is another phosphate group. The third carbon of the deoxyribose is attached to the phosphate group here. The fifth carbon, this one, is attached to the phosphate here. That means the sugar molecule is bound to the phosphate groups in 3-5 direction. In 3-5 direction. This is very, very important. We will be talking about this the next class also, about DNA replication. That's why the direction of the strand is known as 3-5 direction, like this. So the 3-5 direction indicates the attachment of the deoxyribose sugar with the phosphate groups. Now, the phosphate and the sugar, or deoxyribose sugar, they constitute the backbone of DNA. This is the backbone of DNA. The backbone of DNA is made by phosphate and the deoxyribose sugar, this one. This is the backbone. Then, the nitrogen base extends inwards like this. Then, nucleoside. Just I made a mistake here. When the nitrogen base is attached to the deoxyribose sugar, it is known as nucleoside. Nucleoside. When the nucleoside is attached to the phosphate group, it is known as nucleotide. The difference is only one letter. Here S, here T. So, deoxyribose sugar and nitrogen base, they constitute a nucleoside, and all the three, nitrogen base, deoxyribose sugar and phosphate group, they constitute a nucleotide. These nucleotides are arranged one after one like this in a sequence. In a DNA molecule, we find millions of nucleotides. Then, I have told you this is one strand, this is another strand of DNA. The nitrogen bases of the two strands are linked together through their bases, like this. This is the cytosine of this strand, and this is the guanine of the strand. Here we have to remember the rule of complementarity. Cytosine always combines with G or guanine, or guanine combines only with cytosine. Adenine combines only with thymine or pairs with thymine. Similarly, thymine combines only with adenine in the case of DNA. Coming to RNA, I have told you already, thymine is replaced by uracil. If you consider this as an RNA, here you find uracil. But here I am describing a DNA molecule. So, the nucleotides of either side, this is one nucleotide, this is another nucleotide, are joined by hydrogen bonds. These hydrogen bonds are the very weak bonds. Whenever it is required, these hydrogen bonds break up, and the two strands are separated, either for transcription or protein synthesis. Now, see here, thymine is combining here with adenine. Always, thymine and adenine are joined by two hydrogen bonds. This is very, very important. Whereas cytosine and guanine are joined by three hydrogen bonds. Then, there are three types of bonds in a DNA molecule. Two already I have explained. One is about glycosidic bond. It exists between the deoxyribose sugar and the first carbon of deoxyribose sugar. So glycosidic bond is formed between the nitrogen base and the first carbon of the deoxyribose sugar. Then, these are the hydrogen bonds formed between two nitrogen bases, two complementary nitrogen bases. Then coming to the sugar and phosphate. The phosphate molecule is connected to successive deoxyribose sugars. This phosphate group is attached to third group here and fifth group here, or third group here and fifth group here, like that. The third group, fifth group. These bonds, you take the phosphate, there are two bonds. These bonds are known as phosphodiester bonds. Phosphodiester bonds. See here, I have drawn this figure. This joins the phosphate group on one side, and this joins the phosphate group on the other side. So the phosphodiester bonds are two. And these bonds are very stable. It is not that easy to break the phosphodiester bonds. That's why the DNA backbone maintains its structure because of these strong phosphodiester bonds in the 3-5 direction here. So there are we have come across three types of bonds: glycosidic bond, hydrogen bonds, and phosphodiester bonds. Then, the two strands are complementary to each other. The two strands of DNA molecule are complementary to each other. What is the meaning of complementary? Always, thymine joins with adenine and cytosine joins with guanine. Or reverse, vice versa. Guanine with cytosine and adenine with thymine. Here, if you know the sequence of one strand, base sequence of one strand, you will be knowing automatically the base sequence of the other strand. So these are unchanged. The backbone is the same in both. In both the strands, the backbone is similarly constructed. It consists of only deoxyribose sugar and phosphate. The difference is only in the nitrogen bases. So, if you know the nitrogen base sequence of one strand, you will be automatically knowing the base sequence of other strand. See here, if T is here, thymine, automatically what will be here? Adenine. If adenine is here, automatically thymine. If there is cytosine, guanine. If here is guanine, here you find cytosine. So in this way, it is easy to understand what are the nitrogen bases on the other side of DNA strand. And this complementarity is very important in DNA replication and also protein synthesis, and we will talk more about these things later on. So in a nutshell, this is about the structure of DNA molecule. In the next class, let's take up replication of DNA. Thank you.